treating the last subtopic under nucleic acids. We'll be doing section 1.4, which is the last subtopic. We'll be treating protein synthesis. So I explained it before, but I didn't explain it on the slide. So I created a slide so that I can explain it better. Hopefully, if you connect the knowledge that you grabbed at the previous video and this one, surely you'll be done with what protein synthesis, right? So protein synthesis, right? So protein synthesis, it's the process of forming proteins in each cell of an organism to form enzymes, hormones, and new structures for the cells, right? So as you can hear by this word protein synthesis, so it's like protein synthesis means forming or manufacturing. So it's a process whereby like we're trying to form what? Are trying to form a protein, right? So the amino acids will be connecting, they will be connected by what? By peptide bonds, and then they'll be forming what? They'll be forming the protein. So at the end of the whole stage, what we're trying to form here, it's what? We are trying to form a protein, right? So firstly, we'll be starting in the what? In the nucleus and ending in the cytoplasm. At the end of every stage that happened, what our main focus is just to form what? A protein, right? So there are two stages that takes place during protein synthesis, namely transcription and translation, right? So you need to know how to describe these two stages, but mainly the reason I wrote the only transcription, the reason I explained only transcription is because of is the main one that they ask during what? During the exams, right? So they love to say describe the stage transcription. You see, so it's the main one that you need to know. Translation, they don't ask it that much, but you just need to know about it, right? So transcription, it occurs in the nucleus, right? So the double helix DNA molecule unwinds, right? And then when the hydrogen bonds break, the hydrogen bonds, remember, they break because of they have weak hydrogen bonds doing what? Pairing with the what? In between the nitrogenous bases. So those weak hydrogen bonds, they do what? They break. And then the DNA molecule unzip. If you don't want to say unzip, they separate, right? And then one strand is used as a template to form messenger RNA. So remember, we have a strand, we have a complementary strand, right? So a complementary strand is it's a one that forms a what? A template, right? So this process is controlled by enzymes, and this process is called transcription. So every time when you explain transcription, you must just end up with what? This process is called transcription, or start with this process is called transcription. That's a what? A compulsory mark. They start by marking it, and then they review the other work that you wrote, right? So you must always start or end by this process is called transcription. So by the way, I took this explanation from the memorandum. So everything that I wrote here, if you explain it this way, you're gonna get total, right? So translation takes place in what? It takes place in the cytoplasm, right? It takes place in the cytoplasm. So this is the stage of protein synthesis. So firstly, as you can see, the first block there, we have what? We have a DNA molecule. So it does what? Those weak hydrogen bonds, they break and then they form what? A template. And then as you can see, after forming a template, there is a what? There is a messenger RNA that formed there. That's the third block inside the nucleus, and then there's a messenger RNA that forms, and then that messenger RNA, the main focus of it is that it's going to go through the nuclear pore, and that it does what? Um, After going through the nuclear pore, the messenger RNA, it attaches to what? To the site of protein synthesis, which is a what? Which is the ribosome, right? And then after it attaches to it, the remember, the consecutive three bases on a messenger RNA are called codons, and the consecutive bases on tRNA or transfer RNA are called what? Anticodons, right? You need to know the difference. And then what happens after the messenger RNA attaches to the what? To the ribosome? Then what happens? we have the what? We have the amino acids. Those black roundy balls, those are amino acids which connect each other due to the help of what? Due to the help of peptide bonds, and then they end up forming what? They end up forming what we are looking for, which is what? Which is a protein. We are trying to synthesize. We are trying to form a protein at the end of this process. Um, and then that's that, ladies and gentlemen. And remember, translation takes place in what? It takes place in the cytoplasm, and transcription takes place in the nucleus. There are only two sides in the protein synthesis. I hope this video was useful, guys, and thank you for listening. As always, thank you for watching. And up next, since we are done with protein synthesis, we are done with nucleic acids as a whole chapter, so we will be looking at meiosis. Thank you for watching, guys. Have a good day.